This protocol provides a benchtop method for assessing the efficacy and mechanisms of action of histotripsy-aided thrombolytic therapy, or lysotripsy, an alternative, non-invasive approach for treating critical deep vein thrombosis. This technique uses bubble activity to have a two-way effect through an increased fibrinolysis due to an enhanced lytic delivery and the hemolysis of red blood cells within the clot. This benchtop approach to treating deep vein thrombosis facilitates the modeling of blood clots, treatment with lysotripsy, and simultaneous imaging during treatment. Bubble cloud generation, treatment planning, and image guidance can be further used to investigate in vitro histotripsy-based treatments of other diseases, such as kidney and liver tumors. To set up the histotripsy source, mount the source onto a motorized positioning system. Cover the imaging array with a probe cover and fix the array coaxially in the aperture of the histotripsy source. Connect the imaging array to an ultrasound scanning system and completely submerge the histotripsy source and imaging array in the tank. Use a syringe to gently remove any air bubbles, and use the imaging array and scanner to acquire degassed B-mode water images at 20 frames per second. While acquiring the images, adjust the position of the imaging array inside the confocal transducer opening until the bubble cloud is located approximately at the center of the image window and record the detected focal location in the imaging window. Then discontinue the insonation and set the voltage applied to the histotripsy source to 0. To prepare a clot